All right, we're going to talk about the process of meiosis. And meiosis is your body's way of making the sex cells. Meiosis is often described as simply two mitotic cell divisions, and while yes, it's two cell divisions, this is too simplistic of an explanation. Mitosis is happening in the somatic cells, or the body cells, and meiosis is only happening in the sex cells. And while mitosis yields two identical cells, meiosis yields four unique cells, and the fact that they are unique cells is absolutely crucial for our sexual reproduction and for the variation within our species and all the life on the planet, for that matter. Meiosis begins with our normal homologous pairs, and these are in this cell. And we'll look at just a very simple cell, and we have chromosome 1 here, and then its homolog, which we call 1 prime. And then a chromosome 2, and the homolog is 2 prime. And then on these chromosomes are the alleles, and the alleles are the combination of the genes. So for example, we might have an allele for freckles, and so big F for freckles and little f for no freckles. And an allele for a widow's peak, so big W and then a little w for the non-widow's peak, and so on. We begin the process in interphase before the process of division actually begins. And all the chromosomes get doubled, and so now they're copied into sister chromatid pairs and bound together. The number 1 chromosome is with its duplicate, and the 1 prime with the 1 prime, and the 2 prime with the 2 prime, and so on. And so now we have these homologs set up, and we're in prophase. This is when the process actually begins, and they're paired up with each other. So the 1 chromatids are paired with the 1 prime chromatids, and the 2 chromatids with the 2 prime chromatids. And in prophase, this is where crossing over happens, and crossing over is covered more in depth in another video, but in short, there's some mixing and matching of pieces of the chromosomes together. This is a crucial step to add to the variation, so once they're paired up, then they are lined up in the center of the cell, and that's called metaphase, and they line up with their pairs. This is a random assortment of how this is arranged. So we can see here they might be arranged with the 1s on the left side and the 1 primes on the right side for the 1 chromosome, and then the 2s on the left side and the 2 prime on the right side. This could be 50-50 either way. And every time the cell does this, it's just random, whether it's going to put the chromosome on the left, and on the right, or it could be reversed with the 2 primes on the left and the 2s on the right. And then the next step is anaphase. And in anaphase, the chromatids are pulled apart. So the pairs of 1 separate from the pairs of 1 primes, and the pairs of the 2 separate from the pairs of the 2 primes, but they're still in their pairs here. So this is our first cell division. We've now split our 1 cell into 2 cells, and they have the full set of chromosomes. Now we go to prophase 2 and metaphase 2, and it repeats the process. So they're paired up and lined up in the center of the cell again and again in random assortment here. They have 50-50 chance of being swapped left to right or left to right. At this point, because of that random assortment, each time there's a mitotic division, each one is unique because of this 50-50 chance of the ones being on the left side versus the right side, and the 1 primes being on the right side versus the left side. It doesn't look like at this point it's going to make much of a difference, but if you are the baby that comes from that cell, you have a 50-50 chance of getting one version of the gene versus the other version of the gene. And every time this happens, every time your parent makes a new egg or sperm, there's a 50-50 chance of one of these combinations, which is one of the reasons why you share some things with your siblings, but not everything with your siblings, because there's always this chance. And then the last step is that they get pulled apart again. So now each of the 4 cells that are produced is 4 unique cells. And each one only has half the genetic information. Instead of having a full pair of the 1s, a 1 and a 1 prime, for example, or a 1 and a 1, or a 1 prime and a 1 prime, each cell only has 1, the 1 or the 1 prime, and also then has only half of the pair for the 2s, and so on. So if you want to think of it like a pair of shoes, you'll get just the left shoe or just the right shoe. This is crucial because now these are the cells that are the sperm or the egg. And notice they have half the number of chromosomes. But when they then form during fertilization with the other sex cell from the other parent, they'll come together. Then each has half of the pair of chromosomes, so they get the other half of the chromosome pair. And for all of your genes, you always get 2 alleles, either 2 dominants, 2 recessives, or a recessive and a dominant, 1 from each parent. And you get that because each chromosome is carrying 1/2 of those pairs. And so it takes the 2 to come together to give you the full set again. So think of it like the shoe again. You get the left shoe from one parent and you get the right shoe from the other parent. And then by putting this all together, you get 23 individual pieces of chromosome from one parent and 23 from the other parent, giving you 23 pairs or 46 total chromosomes, carrying all your genes. This is what you need to be a fully developed, healthy human being.